How are genetically modified organisms, GMOs, created? Imagine taking a tiny piece of DNA from one creature and slipping it into another to give it new abilities. That's essentially how genetically modified organisms are made. It all starts with identifying the specific gene that controls a trait scientists want to change. This gene can come from almost any organism, including bacteria or viruses, which makes some of these experiments quite unusual. Once the gene is found, scientists use a process called polymerase chain reaction, or PCR, to make many copies of it. They also attach special DNA sequences called promoters and terminators to control when and where the gene is active. Think of these as switches that turn the gene on or off in the new organism. Next, they insert this gene construct into a carrier molecule called a plasmid. Plasmids are small, circular pieces of DNA found in bacteria. They act like delivery trucks, helping the gene multiply inside bacterial cells. After enough copies are made, the plasmid with the new gene is ready to be transferred into the target organism. For plants, scientists often use a natural soil bacterium called Agrobacterium tumefaciens. This bacterium can transfer DNA into plant cells, acting like a tiny genetic courier. Alternatively, they might use a gene gun, which shoots tiny DNA-coated particles into plant cells, or electroporation, which uses electric pulses to open cell membranes so DNA can slip inside. In animals, the process usually involves microinjection, where the DNA is directly injected into fertilized eggs or embryonic stem cells. Sometimes, modified viruses called viral vectors are used to infect cells and deliver the new gene. These viruses are engineered to carry the DNA but not cause disease. Once a single cell takes in the new gene, it is grown into a full organism. In plants, this involves growing cells in special lab conditions until they develop into complete plants. For animals, embryos with the new gene are implanted into surrogate mothers to develop into genetically modified animals. Scientists then run tests to confirm the gene is present, properly integrated, and working as planned. They use techniques like DNA sequencing and PCR to check. The organism is observed over generations to see if the new trait is passed on. This process might seem like a wild genetic puzzle, with DNA pieces from different species stitched together. For example, a crop might carry a gene from bacteria that produces a natural insecticide, or an animal might have a virus gene inserted to study disease resistance. These strange combinations are part of what makes genetic engineering so fascinating and controversial. The results of these experiments can lead to crops that grow in droughts, resist pests without chemicals, or produce medicines. But they also spark debates about safety, ethics, and the long-term impacts of changing life's blueprint.